Хорошо. То есть мне просто говорить и все, да? Хорошо. Спасибо большое, Дмитрий. Спасибо. Благодарю вас. Начинаю. Good afternoon, ladies and gentlemen. Topic of today's lecture for microbiology is viruses. Classification of viruses, structure of viruses, replication of viruses, and cultivation of viruses. History of discovering of the viruses is related to the tobacco mosaic virus. In the photograph on the right part of this slide, you see 4 leaves of the tobacco mosaic plant bitten by the tobacco mosaic virus. In the lower left quadrant of the slide, you see drawing of the structure of this virus. Number 3 demonstrates the rod-shaped virus particles of the tobacco mosaic virus. Option number 1 demonstrates red helical structure of the ribonucleic acid genome of this virus, surrounded by the elements number 2. They are parts of the capsid of the covering that protects nucleic acid from negative substances in the environment. But this tobacco mosaic virus, the tobacco mosaic disease, was studied by the famous scientists. First of them is Dmitry Ivanovsky. Dmitry Ivanovsky in the 1892 year worked in the Nikitsky Botanic Garden in the Crimea on the tobacco mosaic disease. He discovered that filtrability, so-called filtrability, of the tobacco mosaic pathogenic agent through bacteriological filters. It means he demonstrated that tobacco mosaic, that liquids of plants infected by tobacco mosaic disease may be filtered through the bacteriological filters. Size of the pores of these filters usually not more than 0.5 micrometers, and the infectious agents of this virus could pass through these filters and infect healthy plants, causing the symptoms of this disease. In 1899, other scientists, Dutch microbiologist Martinus Bering, discovered the intercellular reproduction of the tobacco mosaic disease in this plant infected by this virus. Martinus Bering used the term virus, introduced this term in the science, to refer to the specific pathogenic molecules incorporated in the cells of the infected agents. And both Dmitry Ivanovsky and Martinus Bering demonstrated that the tobacco mosaic disease causative agent of the disease is very small. Ivanovsky demonstrated that may be filtered through bacteriological filter, and Bering demonstrated that this virus may cause visible under the light microscope intercellular inclusions. But this is history of the virology. When saying about viruses totally, we can to see that viruses are infectious agents that do not possess a cellular organization. What in the painting that you see here, you see the worm-like nucleic acid surrounded by the special coat and lack of the cytoplasm. This is a virus, enveloped virus, that has no cytoplasm and correspondingly has no cell organization. Viruses are not cells. That cannot be seen with the light microscope. Only electron microscope is required to see these very small living forms. Their names may describe. So why different viruses are named by their nowadays names? Because names depends on the structural characteristics. For example, picorna viruses are named picorna because pica, this is a Greek word demonstrated small, RNA, this is a ribonucleic acid. Other viruses are named after diseases they cause. For example, herpes viruses. Herpes virus disease had been discovered before discovery of the viruses causing this disease. It means viruses were named after the disease caused. Adenoviruses are named after tissues where they were first found. Adenoviruses were isolated first from adenoids. Adenoids, they are pathological tissues in the nose of the infected children usually. Geographic region also, geographic region where viruses were discovered. Certain viruses also could be basis for naming one of the Coxsackie viruses, for example, as the Coxsackie. Coxsackie, this is a city name, city situated near to the New York City, in the United States. Also, names of viruses may depend on the means of the transmission. For example, arboviruses are named arboviruses because completely we have to read arthropod-borne viruses. Arthropod, they are insects. It means arboviruses are viruses that may be transmitted from animal to animal or from animal to human being by the mosquitoes or ticks or bites, by what sucking of the blood from the infected host and transmission of this blood to the other. Example is Japanese encephalitis virus transmitted by mosquitoes. Classification of viruses. SHATI2 structure viruses, where size, morphological characteristics, and nucleic acid characteristics serve as the most important classification parameters of the viruses. Also, biochemical characteristics of viruses are important to classify them. For example, structure, mode of replication. Diseases caused also influence the classification of viruses. Encephalitis, for example, this is a disease where brain is infected, and hepatitis where liver is affected by the virus. Classification depends also on the mean of transmission. No, you know that arboviruses, they are arthropodal viruses, and this mean of transmission is related. Other viruses may be transmitted through the air droplets or through the fecal masses infected at least from patient. Also, tissue or organ tropism is important. No, organ tropism, this is a tendency of virus to infect the certain cells of the certain tissue. For example, enteroviruses cause, first of all, diseases of the intestinal tract, but from time to time, these viruses may pass through the blood of patients and infect brain, causing encephalitis. It means enteroviruses are transmitted in the fecal oral route from infected fetus to hands of the people and from hands to the mouth and infect intestinal disease. But in some people, certain percent of people, they were very rare, and this is a transmission through the blood, may infect brain, causing, for example, poliomyelitis. And of course, host-ranged type of host cells important the classification of viruses, but some of them are named animal. Animal viruses are viruses of humans, mice, birds, lizards, and other vertebrates. Plant viruses, for example, tobacco mosaic virus, these viruses infect vegetations, plants, and bacterial viruses named bacteriophages serve the host organisms cells of bacteria. Bacteria are the victims for them. To imagine better classification of viruses, we'll take a look to these 4 viruses. In the upper part of this drawing, you see theoretical structure virus. In the lower, real electron microscopy appearances. So the left virus, left was this rod-shaped virus. This is tobacco mosaic virus, causative agents of the disease that help scientists to discover viruses totally. Of course, viruses that you see are RNA viruses. It means they have RNA genome, ribonucleic acid genome. They are rod-shaped viruses. RNA of these viruses protected by capsid. Capsid consists of capsomeres, or these purple subunits of virus, globules that surround the ribonucleic acid and protect this acid from negative environmental factors. The second virus is adenovirus, virus B. The structure of the virus is different. It's not the rod-shaped virus. This is a icosahedral virus. Icosahedral virus demonstrates the shape in the real microphotography that is near to the spherical. But adenovirus is not really spherical because genome of this virus packed inside the virus particle is surrounded by capsomeres, the protein molecules protecting this genome. A genome of this adenovirus is DNA. This is not the RNA, but DNA virus. And the surface globules of the capsomeres of these protein subunits form the triangle flat surfaces, and complex of these triangle surfaces totally create the structure of the icosahedral geometric structure. The third virus, C, is the influenza virus. Influenza virus is a truly spherical virus, and genome of this virus packed in the central area of the virus particle is RNA. But RNA of the influenza virus, in contrast to RNA genome of the tobacco mosaic virus, consists of from 7 to 8 fragments, fragmented genome. And you see these fragments. Genome consists of the separate elements of these fragments. Some of them altogether constitute the genome. The genome is coated by the protein and by the lipid layer. Lipid layer surrounding the internal virus structure is named envelope and consists of the lipid bilayer. And finally, virus D, bacteriophage T4. In the upper part of the slide, you see that this virus is tadpole virus, consists of head and tail. Tail contains also fibers. Tail fibers help this virus particle to be attached to the bacterial cell, victim cell. After that, tail of this virus contracts and pushes the DNA from the head of this virus through the contracted tail and through the hole of the cell wall of the bacterium to the cytoplasm of this bacterium. In this way, a bacteriophage injects its DNA genome to the bacterial cell. Main structural properties of viruses. Viruses do not have a nucleus of the nucleoid. They are not cells, and the genome of this virus is not named as a nucleus or nucleoid. Viruses have either DNA genome or RNA genome, and never both. Due to this, viruses are subdivided into very large groups: DNA viruses or RNA viruses. Virus that is shown here in the upper part of the slide, the photograph, is the influenza virus. This is an RNA virus, and the genome of this virus is consisting of DNA surrounded by the lipid envelope. Lower you see a theoretical structure of influenza virus. You see that genome consists of the 8 segments of the orange color. This segment surrounded by shell of M protein. M protein is surrounded by lipid. But in the theoretical structure of influenza virus, you see that lipids are shown purple. Spikes on the surface of virus, they are virus glycoproteins, the attachment proteins of the virus that help this particle to be attached to the host cell. Genome of virus, first covering surrounding genome of the virus, DNA or RNA, is named capsid. What is the theoretical structure of the influenza virus? Capsid proteins are shown orange. They form helical structure surrounding nucleic acid. Some viruses have an autolipid bilayer. What in the low part you see of the slide, on the right low part of the slide, you see structure of the coronavirus. You see that red RNA is sheeted to ball-like nuclear protein covering of the virus that constitute the capsid. But capsid from outside is surrounded by the envelope. Envelope is the lipid containing second external coat of the virus particle, and these lipids usually are penetrated by the spike proteins. Here spike proteins are shown as the S proteins, typical for coronavirus is M proteins, some green E proteins, and envelope. This is also presence or absence of envelope, the basis for subclassifying viruses, because some viruses are named envelope. All 3 viruses shown here envelope, and they contain lipids. Many viruses are non-envelope. They contain only capsid, but non-viruses don't have lipid envelope. Reproductive properties of viruses, peculiarities of reproduction. Viruses are so-called obligate intracellular parasites. It means they may replicate only inside of the leaf host cells. They replicate only within cell because they cannot synthesize proteins, and they don't have machinery of the energy synthesis. Viruses, they depend on the protein synthetic machinery of the host cell. Host cell is shown in this photograph that you see here. Host cell is shown brown. Virus particles that are released from this cell by the budding are shown red. But to produce this virus, multiple virus progeny cell gives to this virus reproduction process the cellular energy and cellular protein synthetic machinery, because viruses by themselves don't have these structures. They're not cells. Viruses have unique method of replication. This method suited to infecting of the cell by one virus particle, so-called virion. After that, synthesis of the many molecules of virus nucleic acids and proteins in the host cell, maturation from these components of the progeny viruses, release of these viruses. It means during one cycle that may last 1 day or 1 week, viruses from one maternal virus reproduce themselves to the amounts of hundreds, in some cases thousands of viruses. It means one replicative cycle based on the infecting cell by one virus particle produces hundreds of progeny viruses. This is absolutely atypical for other living forms, for cells. Third point, components of virions are producing the different cell compartments. It means different elements of the future progeny viruses would be synthesized in the cytoplasm proteins, in the nucleus, nucleic acids of the viruses, and the different proteins would be synthesized, some in the Golgi apparate, other
only consuming ribosomes, they're not associated to membranes of Golgi brain. And point 4. Infected cells usually dies after release of newly formed virions. It means host cells usually can withstand virus and die, but in some cases, some viruses may calibrate to the cell, keeping cell alive, and replication of this virus is slow. And due to this, viruses may infect cell a long time. Morphological characteristics, size and shape. No, first of all, size. Host cells, what do you see here? You see in this drawing a fragment of red blood cell. Well, this is a red element of the sphere. This is a part of the red blood cell. You know that red blood cell has the diameter about from 7 to 9 micrometers. In the left part of this drawing, you see, mostly shown by the blue color, different viruses. They have different shape and size, but you see that they are very small. And in the lower part of this drawing, you see fragment, no, half of the cell, Escherichia coli, that is much larger than the viruses. So, host cell is often 1,000 times the volume of the virus that is infecting this cell. Variants shown by the blue color in the size vary from 20 to 200 nanometers. Virus size is measured in nanometers. For example, what tobacco mosaic virus, what you see here, this small rod-shaped virus, is very long, 250 nanometers long, but diameter is small, smaller than 20 nanometers. What adenovirus that is shown in the up left part of this drawing, adenoviruses has a 90 nanometer diameter. Polioviruses, where the smallest virus is shown in the left lower part of this drawing, they are icosahedral and they have diameter about 30 nanometers. Apparently, large viruses are pox viruses, for example, vaccinia virus that has size 300 nanometers to 200 nanometers to 100 nanometers. Now, usually diameter is about 300 nanometers. But Ebola virus that is situated between bacterial cell and red blood cell in our drawing is filamentous, is flexible. The length of this virus may be more than 1 micrometer. This is 1,000 nanometers. Diameter usually about 90 nanometers. So you see that viruses are different in shape and size, and size of virus vary from 20 to 300 nanometers usually when saying about diameter. Shape. Some viruses are spherical, or in the left photograph in the slide you see a red host cell contain many progeny virus particles of the herpes virus. These yellow particles have the diameter about 120 to 150 nanometers, and they are spherical. Mid part of this slide demonstrates the tobacco mosaic virus, the typical rod-shaped virus. And in the right part, you see bullet-shaped rabies virus, bullet-shaped virus. Pox viruses shown in the left part of the slide usually are considered as very large. Their size from 200 to 300 nanometers. And filamentous viruses, Ebola virus, shown in the right part of the slide, they are flexible. Diameter of the virus particles about 90 nanometers. So length may vary from 300 to more than 1,000 nanometers. Viral genome.
Undoubtedly, most important part of the virus is the virus genome. It consists of either DNA or RNA, and it allows us to classify viruses for DNA and RNA viruses. Virus nucleic acids usually are packed into the protein capsid. What is the theoretical structure virus shown here? Capsid surrounds the virus nucleic acids. These both nucleic acids, I mean virus DNA or virus RNA, can be either negative or double-stranded, linear or circular. The RNA can be either single molecule or several pieces of the segmented fragmentary genome that is typical for influenza virus. The RNA can be either of positive or negative polarity, but let's pay attention to this point. What does it mean, polarity of RNA? Only RNA viruses, maybe if do they have single strand RNA, may be subdivided to positive or negative polarity viruses. Positive polarity of the genomic RNA is defined as the RNA with the same base sequence as the messenger RNA. It means that positive sense single-stranded RNA genome of virus serves as the messenger RNA. It means this genomic RNA may directly bind to the ribosomes and start synthesis of the proteins encoded in this virus genome. It means positive polarity genomic RNA of the virus may be translated to the protein directly, and positive sense polarity RNA may initiate start of synthesis of virus proteins that will maintain the future replication of virus RNA. It means is itself positive polarity single-stranded virus RNA is infectious. It means if we inject this RNA only to the cells in the experimental conditions, later we'll see appearance of the completely matured virus particle. In the contrast, negative sense polarity single-stranded RNA has base sequences that is complementary to the messenger RNA. They are not analogous of messenger RNA, but they are only complementary to the messenger RNA. It means negative sense RNA can be translated. Before synthesis of protein, this negative sense RNA genome must be transcribed, and the transcript is the positive sense RNA synthesized on the basis of the negative sense RNA genome as on the template. So, examples of the positive sense RNA genome, this is the genome of the poliovirus. Example of the negative sense RNA genome, this is the genome of the influenza virus that is shown upper as the theoretical structure of this virus. In this figure, you see Ebola virus genome and very, very primitive structure of Ebola virus. What in the upper part of the drawing, you see primitivized Ebola virus particle. Of course, real particle is very, very long, but this is short, and has the hooks of different shape on the ends. But lower, you see the line. This is a Ebola virus genome, subdivided in the portions, and each is a portion marked by the certain symbol, for example, NP, nuclear protein, is marked by the certain color. In the center of this virus particle occurs virus RNA. Virus RNA is intimately surrounded by nuclear protein. Nuclear protein shown here pink, and you see that all the central part of the virus is of the pink color. Nuclear protein also is bounded to the virus protein 35, shown green, and VP24, shown red. At one of the pole of this structure, you see two large molecules of large protein. This large protein is RNA polymerase. And the genome of this protein is the largest, and molecular weight of the genome is very high. In the central part of the genome, you see GP, this is glycoprotein, and you see yellow spike. These spikes are situated on the surface of viruses and serve as the attachment proteins of the virus that attach these viruses to the host cell. Genome of this virus is rather large and consists of the single-stranded negative sense RNA, totaling about 90,000 Dalton in size. This is a rather large genome. 90,000 Dalton. I think this is a very large genome among viruses. When saying about classification of the virus proteins that encoded in the virus genome, we have to say that if the protein is encoded by the genome of the virus and can be synthesized in infected cell, but does not occur in the virion particle, this protein is named non-structural protein. So non-structural proteins, they are proteins that are encoded by virus genome, synthesized in the infected cell, but never packed inside of the virus particle. What in the lower part of the slide you see primitive theoretical structure of virus genome that is surrounded to orange, red, and green elements. Non-structural proteins are shown by the red and green color. They are 2A, 2B, 2C, 3A, 3B, 3C, and 3D. But all these proteins, they're not proteins, but all these fragments of genome calls for proteins that all have this name. These proteins facilitate replication of viruses, facilitate synthesis of the progeny-virus genomes, but they are not components of virus particles. But from all this genome, only proteins VP4, VP2, VP3, and VP1, marked by the orange color, but only these proteins are really structural proteins. These proteins, would take a look to the theoretical structure of the virus. These proteins form the capsid, consisting of these four proteins. Three of these proteins are situated on the surface of this capsid, and one, that is named VP4, is situated in the internal part of the capsid. These proteins are responsible for protection of ribonucleic acid of the virus, with this RNA genome is shown in the helical green structure. And surface components of this virus capsid form the triangle structures consisting of VP1, VP2, and VP3. Well, these triangle structures form all the surface of the virus and give to the virus shape that's characterized as something near to the icosahedral structure. It means non-structural proteins facilitate replication of virus, but are not components of virus particles, and structural proteins, they are present in the virion. What is a particle of the virus that may be released from the one cell and absorbed to the second, causing infection in the second cell. Some viruses contain internal proteins in their core. Some of these proteins are enzymes, such as the polymerases. For example, RNA-dependent DNA polymerase or reverse transcriptase that occurs in the combination to the genome of this virus. What is this theoretical figure structure that you see in this painting? Was this enzyme polymerase is marked as the P64 reverse transcriptase and shown as a small ball that binds to the helical blue RNA of this virus. Some enzymes are proteases, or P10, for example. Proteases serve as an enzyme that cleave so-called polyprotein. This is a very large protein encoded by the virus genome that must be cleaved to the certain fragments to give to these fragments activity normally required during the replication. It means polyprotein consists of all the virus elements, or majority of virus proteins that are associated one to other, and in this state they are not active. Protease of the virus would cleave them to the functional smaller proteins that form the complete normal virus particle. Now we understand that very important classification of the viruses depends on the belonging of the virus, certain virus to the DNA viruses or RNA viruses. 6 families of the DNA viruses now are associated to the pathogens for humans, and at least 13 families of the RNA viruses are known. Very briefly, DNA viruses. DNA viruses have different arrangement and different structure of the DNA. Concerning with the peculiarities of DNA structure, the DNA virus is divided into the single-stranded DNA viruses. It means DNA molecule is one strand of the desoxyribonucleic acid. Там parvoviruses have this structure. Second variant, double-stranded DNA, but double-stranded circular DNA. Papillomaviruses have this DNAs. Papillomaviruses, by the way, are very famous oncogenic viruses of humans that cause tumors. Double-stranded DNA, circular, third variant. Double-stranded DNA, but circular DNA. And one important thing, partially single-stranded, in the contrast to the variant number 2. It means that genome of the virus is a circular DNA molecule one, and the second that is complementary to only part of the first circle. Usually this smaller DNA binds to the third or maybe 50%, third part or half part of the complete DNA molecule. Well, this double-stranded DNA, circular, partially single-stranded, is typical for hepatitis B virus. By the way, also virus causing hepatocellular carcinoma in humans and hepatitis B. And the last variant shown in this table is double-stranded DNA, but double-stranded DNA linear. It means single-stranded DNA, double-stranded linear DNA, double-stranded circular, and double-stranded DNA circular, partially single-stranded DNA. What fourth variants? Double-stranded DNA linear genome is typical for many viruses, for herpes viruses, adenoviruses, pox viruses, and some other. RNA viruses. Structure of the RNA of RNA viruses. First of all, single-stranded RNA positive polarity. Single-stranded RNA positive polarity. Remember that positive polarity RNA means that this RNA may be translated and may serve as a messenger RNA. Such structure is typical for polioviruses. Second variant, single-stranded RNA negative sense polarity. The RNA can serve as a messenger RNA must be transcribed to the positive surface RNA copy to initiate protein synthesis. For this, single-stranded RNA negative polarity virus is typical for measles virus or rabies virus. Third variant, single-stranded RNA virus segmented. This single-stranded RNA negative sense, but segmented. All the genome subdivided to 7 or 8 separate segments, and the complex of these segments in one virus particle serve as the complete genome, but genome coded for these segments. This is a peculiarity of influenza virus, and this peculiarity makes influenza virus very very variable in the genetic and immunological sense, and this variability helps virus to survive in the human populations that have immunity to other variants of influenza virus. The next step, double-stranded RNA segmented viruses with a positive and negative polarity, not single-stranded as the influenza virus genome, but double-stranded. It means positive strand associated to negative strand, and both are segmented. This is typical for rotaviruses. Rotaviruses cause the diarrhea in the young children. And the last variant, single-stranded RNA diploid positive polarity. Single-stranded RNA, but diploid. It means this is typical of human immunodeficiency virus causing AIDS, HIV virus. This virus, in contrast to all other viruses, has not one but two identical molecules of the RNA, and each molecule binds to the own reverse transcriptase, and each RNA molecule may initiate synthesis of the DNA copy of these RNAs. This is a diploid genome. Diploid genome is the distinctive characteristic of all retroviruses. Capsid. Virus capsid. This is a protein code of the nucleic acid. Proteins that intimately bind to the nucleic acids of the virus particle form the so-called capsid. This is a protein code of the nucleic acid. This protein code consists of the capsomeres. Capsomeres, they may be arranged differently, and their arrangement may be responsible for shape of some of the viruses. And you see that in the upper part you see spherical envelope HIV virus, and capsid proteins form the cuneiform structure in the center of the virus that looks like the pupil of the cat. And the contrast, what in the polioviruses shown in the lower part, capsid consists of the capsomeres. Capsomere, the individual global support is associated one to other to form this capsid shell. In the polioviruses, these proteins of the capsid form the icosahedral structure, and what the icosahedral shape of this virus depends on the arrangement of capsomeres. Symmetry of capsid. Capsid of the viruses may have different shape. Capsid proteins interact from one side to the nucleic acid, protecting this side, this structure. From other, capsomeres may combine to the neighboring capsomeres, forming typical structures that are different in different viruses. What arrangement of capsomeres in the structure variants may be described by classification of the variants of symmetry, and symmetry may be helical, maybe icosahedral, and maybe complex. Helical symmetry gives rod-shaped structures of the viruses. If virus has a rod-shaped structure, it means that capsomeres in the capsid of these viruses form tube-like structure with a helical arrangement that is shown as a blue molecule here. In the center of the drawing, you see spherical icosahedral virus. Structure of that consists of the regular elements of the capsid proteins given to this virus shape near to the spherical. Complex symmetry is shown by what structure bacteriophage you see here. Head of this bacteriophage contain nucleic acid inside. This is DNA. Has the cubical or icosahedral symmetry, but tail of the same virus is a cylindrical and of course demonstrates the helical symmetry. And you see that upper part of the virus has the icosahedral symmetry of capsomeres, lower part tail helical. And this is the example of the complex symmetry. Some words about icosahedral or cubic symmetry of the viruses. This variant of the symmetry is demonstrated brightly by the structure of the adenovirus. In the upper part, you see a theoretical structure of the adenovirus. In the lower, real electron microphotography. How are arranged capsomeres of the adenoviruses? They are arranged in triangles, and one of these triangles is shown by the triangle shown by the marker here. You see that all the surface of adenoviruses depends on the many triangles. And with this structure that is near to the spherical, but not really spherical, because surfaces formed from the flat triangles is named icosahedron. What this structure is named icosahedron. Icosahedron structure is approximate outline of the sphere, but not real sphere. Helical symmetry. Helical capsids with the helical symmetry have the capsomeres which are arranged in a hollow coil that appears rod shape or spherical shape. What nucleic acid surrounded by capsomeres with the helical symmetry form the helical structures that pack in the central part of the virus, and if virus is broken, you may see in the debris containing fragments of the virus many capsomeres surrounding nucleic acid released from the virus particle. What do you see here? You see fibers, belt-like structures. External surface of these structures consists of the capsomeres that form hollow tube-like structure. Inside of that is packed nucleic acid. This structure is typical for paramyxoviruses. Autocapsid proteins. Autocapsid proteins have a different role. So, if the function depends on the type of virus. If virus is naked, non-envelope, and doesn't have lipids, autocapsid proteins, in this case, protect the nucleic acid from the inactivation by deleterious agents. One function. They mediate the attachment of the virulence to special receptors of cells. What in the upper painting you see purple receptor of the host cell that the key to the lock very specifically fit to the canyons of the virus. And this is a virus that formed from the autocapsid protein molecules, some canyons that fit to the external surface of the receptor of the cell. And this interaction establishes the beginning of the virus infection, attachment of virus to the host cell. Lower, you see drawing with the poliovirus. This virus shown blue with the pink RNA genome, but surface capsid proteins of this virus specifically interact with the receptors of the nerve cell, shown as the orange structures. With this interaction of the outer surface proteins of the naked viruses to the receptors of cell is responsible for so-called organ specificity of the viruses. With majority of viruses, absolute majority, they specific to the certain organs, not all, but certain, and the specific type of cells in these organs. For example, encephalitis virus may infect nerve cell, neuron, but can't infect skin cell or muscle cell. And the next property, exception for protection, organ specificity, capsid proteins of the naked viruses, non-enveloped viruses, serve as the antigens that generate synthesis of antibodies. But in the low part of the slide, you see interaction of enveloped and non-enveloped viruses to the immune cells, and you see small green, not green, small blue star-like molecules, the antibodies. So virus proteins usually are recognized by the immune cells of human organism that generate antibodies, and these antibodies may specifically react to the viruses, preventing binding of these viruses through the surface of proteins to the host cells. And in this case, antibodies are named neutralizing because they cause effect of neutralization. It means in the presence of these antibodies, virus cannot attach to the host cell, and this antibody prevent development of the disease. Enveloped viruses. Enveloped viruses have the external code, so-called envelope. Envelope shown in this figure, yellow, consists of lipids. The lipids of the host cells that were borrowed from the cell in the process of the maturation and budding of the virus that's shown here. Here is shown influenza virus structure. The outermost covering of the enveloped viruses is the lipoprotein membrane. This membrane establishes envelope. This lipoprotein membrane of envelope composed of lipids, of course, taken from membrane of human cells, and spike proteins. Spike proteins are virus proteins. Here in the influenza virus, you see purple hemagglutinin spike proteins and the blue neuraminidase spike proteins. It means influenza virus has two types of spikes. The processes that are exposed to the outside. Hemagglutinin is the attachment protein. Hemagglutinin, this name is derived from the capacity of this protein to bind to the red blood cells, causing clumping of red blood cells. So this effect is named hemagglutination.
A neuraminidase is the surface enzyme of the influenza virus that cleaves sialic acids of the host cells to facilitate release of virus from the host cells after replication of this virus. Envelope viruses have a matrix protein. It means surface is coated of these viruses by the lipids. We say that they're envelope lipids that in this envelope structure is shaded to the glycoproteins and that form spikes. This envelope surrounds layer formed from the matrix protein. Matrix protein shell is shown here as the purple coat surrounding genome that also is surrounded to the nuclear protein. It means RNA genome, shown red, is coated by nuclear protein, blue in this scheme, and what this nuclear protein complex to the ribonucleic acid is surrounded by the M1 protein. This is a matrix protein that from the surface is surrounded by lipids of the envelope. Presence of the lipids in the envelope viruses make these viruses less or more sensitive to alcohol, chloroform, ether, bile salts, and other lipid solvents. Lipid-containing envelope viruses are sensitive to these solvents. In the contrast, non-envelope viruses that don't have lipids, they're resistant for lipid solvents, and alcohol and chloroform can destroy poliovirus, non-envelope, but can destroy influenza virus, that is envelope. Function of the envelope proteins. Function of the envelope proteins of the envelope viruses are very similar and really the same function comparing to the autoproteins of the non-envelope viruses. So, envelope proteins of the envelope viruses mediate the attachment of the virus to the host cell. Envelope proteins are determinants of the species and organ specificity of the virus. Structure of these envelope proteins regulate by capacity of virus to bind to the certain cell. Envelope proteins of the envelope viruses are the principal antigens against which the host immune response has to be generated. It means antibodies synthesized. It means envelope proteins are the principal, it means most important antigens of the virus. But virus proteins of the envelope viruses are shown in the upper virus that interact in this painting to the two human cell and immune complement molecules. You see that these purple spikes of this envelope virus, they react to the blue antibodies that recognize the antigen of this surface envelope proteins that may neutralize virus. Now, envelope proteins of these viruses are the determinants of the type specificity called serotype of viruses. No, serotype, this is a variant of viruses of certain species that react to the specific antibodies and don't react to other specific antibodies that are specific to envelope proteins of the other viruses or other serotypes. Some viruses have multiple serotypes. For example, influenza viruses have hundreds of serotypes. What does it mean, multiple serotypes? It means all the influenza viruses known are subdivided to strains that are kept in the special collections in the virological laboratories. These strains may vary in their specificity, first of all, specificity to antibodies. But if certain virus strain reacts with a certain antibody,
and other strains don't react to this antibody, it means that this first strain variant of the influenza virus is considered as a certain serotype that is recognized by this antibody that may differentiate this serotype from all other. All other would have no reaction to these antibodies, and correspondingly, all other serotypes also may be detected by the special antibodies that don't cross-react. It means can react to the analogous outer envelope proteins of other viruses. With this variability of the structure of the viruses is named by the term serotypes. Some viruses have only a few serotypes. For example, polioviruses have only 3 serotypes, not hundreds of influenza viruses, but 3. Viral replication. Replication of viruses depends on the synthetic machinery of the host cell and energy producing machinery of the host cell. Viruses have unique method of the replication, where one virus particle infects one human or animal host cell, and this host cell maintains synthesis of hundreds of proteins during one life cycle. Several hundred progeny viruses during one life cycle that may last from 1 to 5 or 7 days. Viruses can cause alteration of cell morphology, accompanied by the degeneration of cell function. Usually, the degeneration causes death of the cell, changing of the cell, the morphological, biochemical, are named cytopathic effects. It means majority of viruses can cause cytopathic effect that demonstrates defects of the host cells caused by viruses. But not all viruses can cause cytopathic effect. Some viruses, for example, rubella viruses, they don't kill host cells but change their function. A virus replication cycle may be subdivided into different phases. First is the recognition and absorption of the virus. The virus is shown on purple to the surface membrane, cellular membrane of the host cell. The second step is penetration. Virus makes cell to form the invagination that after that is transformed to the vesicle, pinocytotic or phagocytotic vesicle, or endocytotic vesicle. All this cytotic vesicle is shown here. After that, endocytotic vesicles contain viruses, contact and fuse their membranes to the lysosomes. Lysosome is not shown in this figure, but lysosomes inject to the endosomes enzymes and acids. And these acids usually, the enzymes, destroy capsid and envelope of the engulfed in this way virus and cause deproteinization or uncoating. What uncoating? Shown here. Uncoating, this is a removal of the virus proteins from the surface of nucleic acid. It means nucleic acid becomes free from nuclear protein, capsid proteins, and all other. And nucleic acid is liberated. It goes to the cytoplasm. After that, this nucleic acid binds to the ribosomes or transcript of this acid if they're transcribed in the host cell, and protein synthesis is initiated. Protein synthesis is part of the step that is named biosynthesis. Protein synthesis is shown in the left part of the scheme, marked by the number 4. Nucleic acid synthesis is shown in the right part of the scheme and marked by the number, symbol number 5. When both proteins and nucleic acids, I mean newly formed proteins and nucleic acids
also newly synthesized, accumulated in the cell, and they may accumulate in different compartments of the cell. Finally, proteins react with the nucleic acids, forming initially capsids, empty. After that, newly formed progeny virus nucleic acids enter to these capsids, forming unmature virus particle. After that, mature virus particle. Maturation, and this is a process of the formation of the functionally active virus particles. After maturation, virus is released from the cell. Usually, these cells, before release, becomes broken, destroyed, because virus synthesis of virus components at the same time cause inhibition of the synthesis of the proteins of the cell and disappearance of the adenosine triphosphate and other energetic molecules. Energy, first of all, is used for synthesis of virus components, and virus suppresses by synthesis of the own cellular substances. That usually causes death of cell. If cell membrane ruptures, we say that this is a lytic cytopathic effect. Some details. First, recognition and absorption. The viral surface proteins attach to the specific receptor proteins of the cells, receptor of the cell. Host cell becomes associated to the virus, stick to the virus. For example, rhinoviruses, what fragment of rhinovirus is shown in the upper drawing, have the canyons, recession in their structure. These canyons spatially fit to the receptor molecules of the sensitive cells of the mucous membrane of the respiratory system. And after that, non-covalent bridges are established between outer proteins of the viruses and receptors of the host cell, and virus becomes attached to this cell. In the lower part of this slide, you see how HIV virus, human immunodeficiency virus, having GP120. GP120, this is lollipop-like surface spike molecule of the HIV, or this GP120 specifically binds to the CD4 receptors of the host cells. These receptors are typical for T helper cells, T lymphocytes of the helper cell population, and for the cells of the macrophage line. And due to this, GP120 becomes binded to the CD40 and initiates process of the penetration of the cell membrane by the virus particle. Penetration. After attachment or binding of virus to the surface of the cell, virus particles usually make cell of producing penocytotic vacuole. And this vacuole contains virus particles inside, migrating the cytoplasm of the cell. So, what this mechanism of penetration of the membrane of the cell by virus is named receptor-mediated endocytosis, or virapexia. In the upper part of the slide, you see electron photograph demonstrating how this endocytotic vacuole is formed. Other viruses penetrate cell not through the endocytosis, but through the fusion. Here you see theoretical mechanism of the penetration of cell membrane by the fusion of the virus lipid envelope to the plasma membrane of the cell, with a release in the cytoplasm cell of the nuclear protein of the virus.
The next step is named uncoating. What in this scheme and some schemes that we will see later, we will see events typical for influenza viruses. It means number one demonstrates attachment of influenza virus to the host cell and begin formation of the endocytotic vacuole. Later, you see that endocytotic vacuole contains virus inside. This process is marked by the number 2. The third event that is shown here, this is event of the destroying of virus particle in the endocytotic vacuole that was not shown but was combined to the lysosome and lysosomal enzymes and low pH acids cause the destroying of the envelope, destroying of the capsid, and release to the cytoplasm of cell of the fragmented virus genome. You see 1, 2, 3, 4, 5, 6, 7, 8 RNA segments of the influenza virus genome. By the way, what's virus genome that you see here released to the cytoplasm, uncoated genome, is a single standing negative cell segmented RNA. You remember, negative strand, negative cells segmented RNA. Then biosynthesis is initiated. Here you see what near to the marker number 2, process of uncoating with the release of the RNA segments of the genome of influenza virus. These segments are transported to the nucleus of the cell. You see a helical structure of the 1 segments of the RNA. This is negative sense RNA and cannot initiate synthesis of proteins. Due to this, negative sense RNA in the nucleus, first of all, is undergo to the transcription. And positive sense RNA transcript, copy of this genomic element, is synthesized in the nucleus. Protein that is responsible for this is RNA polymerase or polymerase protein of the influenza virus. Well, this positive sense transcript serves as a messenger RNA and is transported to the cytoplasm, initiating synthesis of the non-structural proteins. Non-structural proteins are shown in the scheme by the symbols NS1, this non-structural protein 1 of influenza virus, and NS2, non-structural protein 2 of the influenza virus. After synthesis in the cytoplasm of these proteins are transported back to the nucleus and initiate active replication of the minus RNA transcripts from the positive sense RNA, intermediate RNA that was synthesized before. So non-structural proteins serve as the enhancers of the synthesis of the new virus genomes that are shown here, the 1, 2, 3, 4 helical structures. It means by synthesis of the early virus proteins, our this NS1 and NS2 early proteins, is shown by the numbers 3 and 4. These proteins are enzymes which can replicate the viral genome. They known as the virus-encoded polymerases or replicases. This process occurs before viral genome replication. Viral genome replication is shown here and marked by the number 5. It means early. So we have to understand that in the biosynthesis, we have to recognize 3 sub-steps. First, early events or synthesis of early viral proteins. In this case, these proteins would be NS1 and NS2. After that, synthesis of the new genomic RNA molecules of the new genomes. This is a biosynthesis of the RNA transcription phase. And then late events, they're not shown here, but late events would be synthesis of other structural virus proteins that start accumulating in the cell after synthesis of the genomes of the progeny viruses. Let's go. So, what biosynthesis do? So, viral genome replication is shown by number 5 in the scheme. The template nucleic acid, this is a positive sense RNA that was transcribed from the genomic RNA of this virus, is used to produce a full-length negative sense RNA genome. With this full-length negative sense RNA genome, variants are shown by these 4 molecules. And late mRNA for the synthesis of late circular proteins, structural proteins. It means with this positive sense RNA serve to both synthesis of new genomes and synthesis of the late phase proteins that are M protein, hemagglutinin, neuraminidase, and other structural proteins of the virus. Biosynthesis of late virus proteins. What late virus proteins are shown as the PB1, this is a protease B1, protease B2, nuclear protein M1 protein, M2 protein, and hemagglutinin and neuraminidase. So, the late events are demonstrated by synthesis of late virus proteins, structural proteins. The late events occur after synthesis of viral genome, and result in the production of structural proteins needed for building capsids and envelopes. What for? The arrow shows for this process of the accumulation of the virus proteins, structural proteins. The capsid virus proteins, the N nuclear protein, NP, is translated first, and the envelope glycoproteins, hemagglutinin and neuraminidase, synthesized in the endoplasmic reticulum and Golgi parade, are then synthesized. These proteins, hemagglutinin and neuraminidase, are processed. It means they go into the cleavage by polymerases, and then becomes functionally competent and play important role in the assembly of the virus particles. Maturation and assembly of virus particles. Progeny virions, what here you see moment of the budding of the progeny virions, can be assembled from the replicated viral genomic material or genomic negative sense RNA and newly synthesized capsid protein. Notice newly synthesized capsid protein, this is a NP protein. NP protein is shown as a small, what's in the scheme, small ball surrounding genomic RNA. Expenditure of energy for packaging the virus genome within the capsid protein is not required. The capsid proteins, or these capsid proteins surrounding the newly formed nucleic acids, they assemble to the genomic RNA and then associate with regions of plasma membrane from inside, contain envelope glycoproteins. No, really, what is happened? Glycoproteins are membrane proteins. They, after synthesis to the chambers of Golgi parade, are exposed on the surface of the plasma membrane. Transmembrane elements of these glycoproteins have the small portions that are exposed to the inside of the cytoplasm of the cell. But these portions interact with the newly synthesized M1 protein, matrix protein, that forms the layer that is formed by the contact from M protein to the hemagglutinin and the neuraminidase
transmembrane portions. After that, this M protein reacts to the nuclear protein associated to the RNA, attract these newly formed RNAs to the certain part of the cell membrane, and in this way buds are formed. Buds consist of the attracted to the series progeny virus RNAs associated to newly formed nuclear proteins associated to M protein. This is a packed in the newly formed structure, but is detached from the cell and forms a new virus particle. Release of progeny variants. The progeny variants of the naked viruses are released via a rupture of the cell membrane. This rupture of the cell membrane is due to disorganization of the skeleton structure of the cell and finishing inhibition of the protein synthesis of the proteins of the cell and synthesis of lipids of the cell with the rupture of the plasma membrane and release of newly formed viruses to the outside. Cell usually dies. Envelope viruses are released differently. Envelope viruses bud, and this budding has been described before in my lecture. You see here the upper painting formation of bud. Analogous process is shown in the lower photograph when some buds are almost completely matured, but some buds only start to be formed. Here you see most important steps of the replicating cycle of the enveloped herpes virus. Herpes virus attaches to the cell, host cell. After that, fusion of the envelope of this virus to the plasma membrane is happened. Nuclear protein of the virus is injected to the cytoplasm. Proteins surrounding nucleic acid of the virus lysed. Nucleic acid is transported to the nucleus. In the nucleus, process of the transcription for this nucleic acid, this is DNA, is happening. mRNAs specific for viruses are transported to the cytoplasm. Here, initially early proteins are synthesized. After synthesis, migrate back to the nucleus, start support process of the formation of new virus DNAs. After the synthesis, new portion of mRNAs is transported to the cytoplasm and late virus proteins are synthesized. After the combine of the newly formed virus nucleic acids and proteins is happened, viruses start to be released by budding from this virus to the extracellular space. Isolation, cultivation, and detection of animal viruses. No, this slide and other would be devoted to the laboratory diagnosis of viruses and maintain them in the laboratories. Mainly three approaches can be used for the cultivation of the viruses. First, animal inoculation. Second, hibernated eggs inoculation. And third, cell culture inoculation by the viruses. Animal inoculation first is the earliest method for virus cultivation. This method is based on the injection of the virus-containing materials to the laboratory animals, among them primates, guinea pigs, rabbits, ferrets. Mice, until now, are most common animal that is used in the attempts to maintain viruses after animal inoculation. Replication of viruses in animals may cause death, disease development, and formation of visible lesions. Detection and registration of all these things are important to understand are viruses, do viruses capable of replicating in this animal or not. In this slide, you see cultivation of animal viruses in the embryonated hen's eggs. You see that what 4 injections may be done to the horizontal cavity that's shown pink, to the amniotic cavity, amniotic liquid that surrounds the embryo, to the yolk sac, and to the embryo directly. Embryonated hen's eggs may maintain synthesis of viruses, and development of infection may be demonstrated by the formation of visible lesions in the hydroaltoic membrane of the egg. What here you see hydroaltoic membrane of the egg embryo after removing of the embryo, and what white dots. They are places of the cytopathic effect with the lysis of cells, and they are named POX. POX because what these defects are caused by pox viruses and some other viruses. This photograph, a photograph revealing smallpox virus pox on the hydroaltoic membrane of the virus is shown. Viruses may be accumulated in this case in the not only in the depth of the hydroaltoic and other membranes of the embryonated egg. Viruses may accumulate in the liquid materials, in the hydroaltoic liquid, in the amniotic liquid, and may be isolated from this liquid and may for the further investigation. So here you see inoculation of virus into amniotic sac, or amniotic sac. This is access to replicate influenza viruses. Viruses may be inoculated to the allantoic cavity. This also is used to accumulate influenza viruses, or yolk sac. For the yolk sac, usually accumulated such viruses as Coxsackie viruses. How viruses may be detected in the liquids of the embryo, for example, in the hydroaltoic membrane. One of the methods is named hemagglutination, that may be applied for hemaggglutinating viruses that have proteins hemagglutinins that may attach viruses to the red blood cells. When red blood cells, are added to serial dilution of the viral suspension, virus-containing materials, for example, from 1 to 3, for example, you see serial dilutions by 2 with the step, with the dilution in each what transfers the material with the extent 2. We see that after adding to this diluted material of the red blood cells, they may cause hemagglutination effect when virus is highly diluted and demonstrate no activity. You see sedimentation of red blood cells to the bottom of this U-bottom plastic plate, bottoms of the bottom plastic plates. Hemagglutination titer in this case, this is a characteristic of the concentration of virus. Here, titer is shown in the tube number 8. It means that what dilution made by the A24 transmission of the material from one volume to other, what demonstrate what this titer that is typical for the tube 8. What in the row B, titer would be 3, because in the first and second tube something is seeing agglutination, but after that you see only normal sedimentation of red blood cells on the bottom. Why this effect is achieved? What in the left part of the slide you see that hemagglutinin viruses, after mixing to the red blood cells, form the large complexes, and these complexes sediment not to the deepest, most deep place of the U-bottom plastic tube in this plate, but spread over all the curved surface of the tube, and the effect of the small pellet is absent. The effect of inverted umbrella. The second method of cultivation of virus is the tissue culture. Tissue culture is the culture of bits of tissue or organ in vitro. It means external to the animal or embryotic organism. Growth of the virus in tissue culture is the most important method for the cultivation of viruses. There are three types of tissue cultures known. One is organ culture, second explant culture, third cell culture. All these things are subtypes of the tissue cultures. First, organ culture. For example, tracheal ring organ culture. Tracheal ring organ culture, isolated from mice, for example, or rats. Tracheal ring organ culture, the example of the organ culture, is used for isolation of coronaviruses. Explant cultures. Explant cultures, they're pieces of organ isolated from the certain tissues, mostly pathological. For example, from adenoid tissue, explant culture that is used for adenovirus application. But most important subtype of the tissue culture is named cell culture. Cell culture, this is a growth of the viruses on the suspension of cells isolated from the organ and occurring in the form of the monolayer or suspension, not in the fragment of the organ or in the small pieces of the tissues after cutting by the special instruments. Cell culture is most important. Now, cell cultures. Cell cultures consist of separated cells. They may be classified into three types on the basis of what now I say, not about organ cultures, but only about one subtype, subtype of organ culture that is named cell culture. What are the distinctive characteristics of this? Cell culture consists of separate cells. They are not fragments of organ. They are not pieces of organ, complexes of cells cut it from organ. They are suspension. Cell culture is classified into three types on the basis of origin, what the cells are taken, animal, human, bird, and other. Second, cell culture is classified on the basis of number of generations through which they can be maintained. It means some cells can live longer time, 1 or 2 months or 3 months, some very short, 2 weeks, some may live unlimited time. It means subcultivations of this third type of cells is very long lasting. And third characteristic of cell culture is the chromosomal characters. Are they changed or not? Karyotype, is it changed or not? So, cell cultures are subdivided to primary cell cultures, diploid cell cultures, and continuous cell cultures. First, primary cell cultures. Primary cell cultures are normal cells, freshly taken from the body and cultured. They are capable of limited growth and can be maintained in serial cultures. It means primary cells cultures are named primary because in every new experiment we have to take freshly prepared primary cell cultures. Keeping of these cultures in the laboratory for a long time is impossible. Examples: rhesus monkey kidney cell culture, produced from explant
taken from the kidney of these monkeys. Second example, human amniotic cell culture. Human amnion cell culture. No, human amnion cell culture may be taken after delivery. And third, chick embryo fibroblast cell culture. So bird embryos, where we take not the entire embryo, but isolate from the chorioamniotic membrane fibroblasts and manipulate with these fibroblasts later. In this scheme, you see how primary cell cultures are prepared. They may be prepared from the mouse, mammals, embryo, hibernated eggs. But here you see organ of the maybe monkey. This very typical source for cells. The explant of this organ is uptaken. Then this explant is placed to the solution of the proteolytic enzyme, trypsin, for example, with a lower concentration. Trypsin destroys bridges between cells in the pieces of the organ that are placed here. No, before exposure to trypsin, this explant, of course, is cut for small fragments, and red blood cells are removed, and from the pieces of organ, trypsin isolates separate cells, because trypsin irreversibly destroys the bridges between neighboring cells in the organ, and these cells become free and accumulate to the bottom of the plastic plate or glassware. And you see finally what these cells. You feed these cells by the special growth media, and they maintain during a certain time. Diploid cell cultures. Diploid cell cultures are cells of the single type that retain the original diploid chromosomal number and karyotype during serial subcultivation for limited number of time. It means their karyotype may be maintained during 20, 30, or 40 serial passages. One passage lasts 3 or 5 days, where you give to cells new fresh growth medium. But if you will give them more than 50 serial passages, cells will undergo to senescence. They will die. Examples: human and animal embryonic fibroblasts. So normal, but fibroblasts, usually embryonic. Examples: WI38, this is human embryonic lung cells, and HL8, rhesus embryonic cells. And the most practical, continuous cell cultures, or continuous cell lines. They are cells of the single type that are capable of continuous serial cultivation during many years. These cells may practically admit unlimited potential for growth. The cells may be maintained by serial subcultivation or stored in the cold at the -70 or -80 Celsius. Examples. Now, first of all, what are the source of these continuous cell cultures? They are not normal or amniotic. They are cancer cells. What, for example, HeLa. HeLa cells, they are human carcinoma or cervix. This cell culture lives maybe about 60 or 70 years until now. HEP2, HEP2, human epithelioma of larynx cells. And third, KB, human carcinoma of nasopharynx. In the photograph you see HeLa cells are the Hemsey stain of these cells. What these cells with very small droplets inside, they're cells of this tumor line. How these tumor cells are prepared? From the tumor after cutting of these cells and trypsinizing, treatment by trypsin, expression of the cell is cultured on the bottom of the plastic jar, for example, plastic vial. And after that, you try to recognize various focuses of the growth. Normal cells usually form the monolayer and suspended. Tumor cells, they form the conglomerates, elevated conglomerates consisting of many cells. After special stain of these conglomerates, you may see colonies consisting of tumor cells. What in the left part of the slide is a photograph of the HeLa cell culture. HeLa. Detection of virus growth in cell cultures. Detection is based on many axes. One of them is registration of cytopathic effect. Cytopathic effect caused by different viruses may be different. For example, cell rounding, typical for herpes simplex virus type 1. Shape of cell is changed, they becomes round. Cell lysis. What enteroviruses are very lytic viruses. What in the photograph you see normal Vera E6 cells in the left part of the photograph, and the same cells after infecting by SARS coronavirus. Third, inclusion body formation, accumulation of the fragments of the viruses in the certain parts of the cell that may be stained specifically. Cytoplasmic localization, when some cells becomes enlarged and cytoplasm becomes filled with vacuoles. And last, ABCDE variant, syncytium formation. Syncytium, this is a result of fusion of many cells in one multinucleated conglomerate. Here you see how cytopathic effect is caused by sarcoma virus, Rous sarcoma virus. Rous sarcoma virus causes cytopathic effect that is realized as the tumor transformation of cells. On the left part of photograph you see normal fibroblasts of the chicken fibroblast. In the right part you see formation of the change in the shape tumor cell, because sarcoma rous is oncogenic virus. Localization. In this cell culture you see normal cells, they're fibroblasts, they are shown in the gray long cells. Two cells here are vacuolated. These cells are shown in the right part. You see that nuclei of these cells are surrounded by large cytoplasm, larger than in the other cells, that is filled with the vacuoles, many vacuoles. What formivirus? This is one of the retrovirus, human formivirus, may cause this vacuolization of cells. About inclusion body formation. Inclusion bodies may be stained, developing culture, color that is different from color of the cell. Inside of that, these inclusion bodies are formed. What here you see low magnification, high magnification microscopy of the cells infected by the rabies virus. On the left part you see red dots. They are inclusion bodies consisting of the accumulated capsids of the virus. Capsids and nucleocapsids of the virus, non-functional, defective. In the right part you see only one cell, one purple cell with a blue, dark blue nucleus and round red inclusion body where these virus elements are deposited. Here you see syncytium formation. Syncytium, this is a result of fusion of many infected cells to one cell conglomerate. with the common cytoplasm, very large, and many nuclei trapped inside of this conglomerate. Syncytium, on the left you see syncytium caused by measles virus, on the right syncytium caused by respiratory syncytial virus. Virus name of the species of the virus contains this word syncytium, respiratory syncytium virus, RSV. Our the syncytium of these viruses. In the working with the hemagglutinating viruses, in cell cultures, viruses may be detected by treating on the infected cell culture by the red blood cells. Red blood cells bind to the surface of virus-infected cells, and you see aggregates of red blood cells over the cytoplasm of virus-infected cell. What hemabsorption? This effect is named hemabsorption. No, what measles virus, influenza virus may cause this effect. Immunofluorescence assay is widely used to demonstrate by immunofluorescence depositions of virus antigens. But here you see cell culture infected by cytomegalovirus. Cells shown red or orange are cells with the virus materials inside are shown as the yellow depositions of the virus antigens. Here you see herpes virus. This is also cell culture infected by herpes viruses. Herpes viruses are antigens of this virus accumulated in the area of the yellow color. What here, here. Green are the normal elements of cells or normal cells. About this yellow, they are very concentrated depositions of these viruses. Now, few words about bacteriophages. Bacteriophages are viruses that may parasitize on the bacteria. We here see bacterial surface with these filaments of flagella maybe, and what bacteriophages attack the cell by the fibers they recognize specific receptors on the cell. Viruses bind to the surface of the cell and penetrate cell wall by the enzymes that accumulated on the basis of the tail. After that, virus contracts its tail and DNA of virus becomes injected to the cell. After finishing all the stages of replication, bacterial cell dies and becomes ruptured. And you see here this fragment of ruptured cell. Progeny viruses are released, some hundreds of progeny viruses, to the extracellular growth medium, infecting other cells. Here you see different shapes, classification on the basis of shapes of the different types of bacteriophages. You see that mostly they have icocidal structure, but tail may have different length, may be replaced by the capsomeres on the vertices. Some phages have the rod shape characteristics, some spherical. Here you see replicative cycle of the virus. Virus infects cells, injects DNA to the cell, DNA replicates, virus proteins accumulated in the cell, virus maturation is happening, and after that, 25 minutes later, viruses, bacteriophages are released, causing death of the cell. This is a lytic life cycle, what's shown here, lytic, causing lytic effect. What here, lytic cycle is shown in the upper part of the slide. Virus is released from the cell, infect the next cell, and next cell becomes infected and may be killed with the formation of viruses. But some infected cells integrate virus DNA in the own chromosome of the bacterium. This is typical, for example, for Vibrio cholera, Shigella dysentery. This integrated DNA doesn't cause the active biosynthesis of the virus components. This integrated virus genome may occur in the dormant state during rather long time. When this infected cell, that is named lysogenic cell, life cycle where lysogenic cells are formed is named lysogenic cycle, this lysogenic bacterium later will replicate, will synthesize second genome, and this genome would be associated to virus integrated genome also. These genomes will be situated in different parts of the cell during the replication of the cell, and after cross-wall formation, two maternal bacterial cells would be formed, and every of the cell will contain integrated virus genome. This cycle may be very long, may last for many years, but in a certain period of time, these lysogenic cells, after contact with a certain activator, so UV light factor that will activate virus genome, may be transferred to the lytic cycle. And with this virus bacteriophage genome becomes detached from the genome of cell, actively replicates, killing the cell with the formation of new viruses, new bacteriophages. Lysogenic cycle with the integrated virus genome. Now, this is the last slide in my today's lecture. Thank you for coming to my lecture. Of course, only 2 students came, but I hope that this lecture will be helpful to you in the better understanding of the virology. Thank you very much. Goodbye. Да, я все закончил. Спасибо, Дима. Я просто повыключал их, да и все. Ну все, спасибо большое. А какие-то новости есть? Вы говорили, что собрание там какое-то будет. Чтобы не отъезжали. Ну, они боятся войны, конечно, это понятно. Ну, Дмитрий, это же нормально. Собственно, если обучение имеет дефекты, то и какие-то там изменения, то это же всегда предмет торговли, это же бизнес. Ну, понимаете, когда вы приезжаете в страну, которая там спокойная, вы там рискуете мало, а потом вдруг раз оказалось, что там в 200 километрах от вашего университета идет война, или даже меньше, чем в 200. Ну и их можно понять. Ну ладно, я не об этом, собственно. То есть пока наша администрация пытается удержать их здесь, да, я так понимаю?
Ну, это они еще обсудят. То есть наши же захотят не потерять деньги, так-то они уедут и что? И все. Ну, наверное, наши попытаются не на дистанцию их перевести как-то. Это же уже было на фоне эпидемии, когда фактически все поголовно студенты находились на дистанционном режиме, там, ну, долго находились. Ну, вы ж, как и мы, все это пережили. Ну, хорошо, спасибо, Дмитрий, спасибо. Ну как, ко мне пришло аж целых 2 студента. 2 человечка всего. Ну ладно, послушаем, что нам скажут наши руководители дальше.